To mount an effective immune response, a host must first identify the threat. Antigen-presenting cells, or APCs, continuously sample the environment, capturing fragments of pathogens and delivering that data to B and T lymphocytes. This logic diagram illustrates the two primary processing tracks. On the left is the endogenous pathway. When a virus replicates inside a cell, proteasomes degrade internal proteins into peptides. These fragments are loaded onto MHC class I molecules and translocated to the cell surface for recognition by CD8-positive T cells. The exogenous pathway handles extracellular threats. APCs ingest bacteria into phagolysosomes, where they are digested into peptides. These peptides bind to MHC class II molecules and are presented to CD4-positive T cells to coordinate the broader immune response. While the biological end goals are nearly identical to mammalian models, the avian immune system achieves these results through a distinct genetic and developmental toolkit. Tracking this system requires following the two primary avian APCs, macrophages and dendritic cells. We can trace their journey from bone marrow progenitors into fully operational, mature sentinels. Understanding avian immunity requires mapping the specific biological functions of these cells and the unique laboratory models and cytokine triggers required to study them. Avian tissue macrophages and dendritic cells originate from circulating monocytes, which are themselves derived from bone marrow myeloblasts. This differentiation is regulated by specific cytokines. Myeloblast proliferation is driven by chicken myelomonocytic growth factor, or CSF3, which acts as the initial catalyst pushing cells toward the monocytic lineage. Once these cells enter the tissue, their final maturation into macrophages depends on CSF1 or interleukin-34, binding to the CSF1 receptor. Researching these cells in birds presents a unique challenge, a significant lack of the comprehensive monoclonal antibody toolkits available for mammalian models. To bypass this limitation, researchers rely heavily on in vivo elicitation methods to source primary cell cultures directly from the bird. For example, injecting a Suffidex suspension into the peritoneal cavity forces a rapid influx of inflammatory macrophages, which can then be harvested and quantified. Beyond primary cultures, researchers employ a second critical tool, retrovirally transformed cell lines. The HD11 cell line is a prominent example. Created via V-Mic retroviral transformation, it serves as a consistent standard for studying avian macrophage behavior. Microscopic evidence illustrates how these different sourcing methods physically manifest in the laboratory. In panel A, we see monocyte-derived primary macrophages. They are uniform, relatively small, and remain loosely adherent in their non-activated state. Contrast this with the HD11 cells in panel F. The transformation results in a distinct, slightly adherent morphology that differs from the primary cellular baseline. Despite these morphological shifts, HD11 cells reliably replicate vital effector functions like oxidative bursts and Fc receptor-mediated phagocytosis. These specific culture techniques provide the foundational lens through which avian myeloid biology is decoded. In the tissue environment, mature macrophages monitor for pathogen-associated molecular patterns, the structural hallmarks of invading microbes. While mammalian immunology uses various markers for activation, the avian model relies heavily on one specific biochemical readout, the production of nitric oxide. Detection of a pathogen triggers the avian macrophage to produce the inducible enzyme nitric oxide synthase, or NOS2. This pathway maps the conversion. NOS2 catalyzes the transformation of the amino acid L-arginine into L-citrulline and highly reactive antimicrobial nitric oxide. This reveals a critical constraint in chicken biology. The birds lack the specific urea cycle enzymes required to synthesize L-arginine internally. Consequently, the ability of an avian macrophage to produce nitric oxide is directly dependent on the bird's dietary intake of L-arginine. Researchers track this activation in the lab by measuring nitrite, the stable byproduct that remains after nitric oxide rapidly degrades. The Grice reaction is the standard assay used to detect these nitrite levels in the culture medium. Using this assay, scientists identified specific microbial patterns and confirmed that chicken interferon gamma acts as a highly potent activator for these macrophages. These chemical activators produce visible physical changes in the macrophage population. Panel C shows the baseline. These are non-activated macrophages, which appear rounded and relatively small. In panels D and E, the cells have been activated by interferon gamma. They have flattened and expanded, showing visible phagocytic activity as they ingest yeast cells. Panel F displays a different morphological footprint, triggered specifically by exposure to bacterial lipopolysaccharide. Each stimulus yields a distinct morphological and biochemical response that researchers can quantify via nitric oxide output. Nitric oxide synthesis serves as both a lethal antimicrobial weapon and a reliable experimental proxy for the avian innate immune response. Macrophages exist to destroy pathogens locally, but dendritic cells have a different endgame, initiating the broader adaptive immune response. To bridge these two systems, an avian dendritic cell must undergo a functional shift known as maturation. In its immature state, the dendritic cell is optimized for antigen capture and sampling. This is reflected in the cell's activity levels. On the left, we see high rates of endocytosis and phagocytosis, mediated by dense mannose receptors on the cell's surface, a state identified by the KUL01 antibody. When the immature cell encounters a stimulus like LPS or a CD40 ligand, it begins a massive phenotypic inversion. The cell shifts into a mature state. It intentionally shuts down its capture machinery, ceasing phagocytosis and significantly reducing endocytosis. This capture machinery is replaced by a high density of presentation and co-stimulatory molecules. Surface markers like MHC class II, CD40, CD83, and DEC205 spike, preparing the cell to communicate with T cells. Researchers verified this shift functionally using a mixed lymphocyte reaction, or MLR. In this assay, only fully mature dendritic cells possess the co-stimulatory density required to drive the proliferation of naive T cells. Mature avian dendritic cells successfully drive robust T cell proliferation in both allogenic and syngeneic reactions, confirming their functional competency. This maturation represents a complete functional redirection. The cell ceases the indiscriminate capture of debris, becoming a specialized communication hub for the adaptive immune system. The final logistical challenge is location. Unlike mammals, birds do not possess a conventional network of lymph nodes. An immature dendritic cell that captures an antigen in the skin or lung is often anatomically distant from the naive T cells it must activate. Maturation triggers a migratory response to bridge this gap. The cell downregulates the CCR6 chemokine receptor and upregulates CCR7. This shift acts as a homing beacon, signaling the mature cell to migrate to the spleen or specialized dermal lymphoid nodules. This sequence defines the cell's life cycle. A progenitor divides in the bone marrow, settles into tissue as a chemical sensor, and upon detecting a threat, physically reconfigures to transport antigen across the body. The avian antigen presentation system represents a compelling example of convergent evolution, delivering a sophisticated immune response through an entirely distinct lineage of cytokines, markers, and cellular logistics.